Welcome to this lecture on the basics of neuroscience. Today, we are going to explore one of the most fascinating and crucial structures in the human body, the blood-brain barrier. Imagine your brain as the most exclusive VIP club in the world. It needs a flawless security system to keep the chaos of the outside world at bay. By the end of this video, you will understand exactly how this microscopic security team operates, why human life would be impossible without it, and the mathematical principles that govern what gets in and what stays out. Let us dive in. To begin our journey, let us build an intuitive understanding of what this barrier actually does. Think about the blood flowing through your body right now. It is like a busy, chaotic street. On that street, there are vital nutrients and oxygen that your brain desperately needs to function. However, circulating right alongside them are dangerous toxins, bacteria, and fluctuating chemicals. If these hazards entered the brain, they would cause absolute disaster. To keep the brain safe, the body employs a microscopic, highly trained team of bouncers at every single entrance. This strict security system ensures that only the right molecules get into the brain, while the dangerous ones are kept firmly locked out. Now that we understand the VIP club analogy, let us look at the formal biological definition. In scientific terms, the blood-brain barrier is a highly selective, semi-permeable border of cells. It physically separates the circulating blood from the brain and the extracellular fluid in the central nervous system. In the rest of your body, blood vessels have tiny gaps that allow substances to leak out freely into the surrounding tissues. But the brain is different. The blood vessels in the brain are tightly sealed, creating an impenetrable wall. This structural difference is the very foundation of the brain's defense mechanism, preventing unwanted substances from simply slipping through the cracks. Building on that anatomical foundation, let us examine the specific cells that create this impenetrable seal. The walls of these blood vessels are made of endothelial cells. Unlike normal vessels, these cells are stitched tightly together by structures known as tight junctions. But they do not work alone. These blood vessels are wrapped by the foot-like extensions of star-shaped support cells called astrocytes. Astrocytes play a critical, active role. They constantly send chemical signals telling the endothelial cells to maintain their tight seals. Together, the endothelial cells and astrocytes form a physical and metabolic barrier that strictly regulates exactly which ions, molecules, and cells can pass into the delicate environment of the brain. Having discussed the cellular components, let us now visualize them together in this diagram. What you are looking at is a cross-section of a blood vessel inside the brain. Notice the central tube where the blood flows. The wall of this tube is formed by endothelial cells, and you can clearly see the tight junctions locking them together. Wrapped around the outside are the blue, star-shaped astrocytes, using their end feet to grip the vessel. Look at the arrows. Essential molecules like oxygen and glucose are allowed to pass through the barrier to feed the neurons. However, notice the red toxin molecule. Its path is blocked by a red X, demonstrating exactly how this physical barrier stops dangerous substances from entering the brain tissue. Now that we have seen the structure of the barrier, we must ask why such extreme security is necessary. The importance of the blood-brain barrier cannot be overstated. Human life would be impossible without it. Your brain is made of neurons, which communicate using very precise electrical and chemical signals. This communication requires a perfectly stable environment. If the barrier did not exist, A simple meal could flood your brain with amino acids and hormones. These would act as false signals, potentially causing massive seizures or permanent brain damage. Furthermore, the barrier protects the brain from deadly infections and circulating toxins that the rest of the body's immune system might easily fight off, but would be fatal to delicate brain tissue. While this incredible protection keeps us alive, it leads us to a massive real-world problem. Because the blood-brain barrier is so good at keeping foreign substances out, it actually becomes a huge challenge for doctors and pharmacologists. When a patient develops a brain tumor or a severe brain infection, doctors need to deliver medication directly to the brain tissue. However, the barrier stubbornly prevents most life-saving drugs from entering. The bouncers do not know the difference between a harmful toxin and a helpful medicine. They just see an unauthorized molecule and block it. Therefore, medical researchers must constantly find innovative chemical ways to trick the barrier or bypass this strict security system to treat neurological diseases. To solve this medical challenge, we must understand the physics of how substances actually cross this barrier. We can quantify this using a simplified version of Fick's law of diffusion. The formula is J equals P times A times the difference between C blood and C brain. Let us break down every symbol. J represents the net flux, which is the total amount of a substance entering the brain per second. P is the permeability coefficient, measuring how easily a molecule slips through based on its size and lipid solubility. A is the total surface area of the blood vessels available for exchange. Finally, C blood and C brain are the concentrations of the substance in the blood and brain. Physically, if you increase the permeability, P, or if the concentration in the blood is much higher, the total flux, J, will increase significantly. Now that we have our formula, let us see this with a concrete example by calculating the delivery of glucose, a vital sugar. We are given a permeability coefficient of 2 × 10⁻⁵ centimeters per second. The capillary surface area is 100 square centimeters. The concentration of glucose in the blood is 5 milligrams per cubic centimeter, and in the brain, it is 1 milligram per cubic centimeter. We want to find the net flux. Our first step is to find the concentration gradient, which is the driving force for diffusion. We subtract the brain concentration from the blood concentration. 5 minus 1 gives us a difference of 4 milligrams per cubic centimeter. This positive number means glucose naturally wants to flow into the brain. With our concentration difference calculated, we can now find the final flux. We take our main equation, J equals P times A times delta C, and plug in our known values. We multiply the permeability coefficient of 2 × 10⁻⁵ by the surface area of 100, and then by our concentration difference of 4. Multiplying the area and concentration gives us 400. Finally, multiplying that by the permeability gives us our result. The final net flux is 8 × 10⁻³ milligrams per second. Physically, this means that every single second, exactly 0.008 milligrams of glucose successfully cross this specific section of the barrier to feed the hungry neurons, keeping your brain functioning properly. Let us apply this to the medical challenge we discussed earlier. Imagine you are a pharmacologist designing a new drug. The capillary area is 50 square centimeters, and the concentration difference is 10 milligrams per cubic centimeter. Your initial drug is not very lipid soluble, so it has a very low permeability of 1 × 10⁻⁷ centimeters per second. We need to find out how much of this old drug is reaching the brain. We multiply the old permeability by the area and the concentration difference. This gives us a very tiny flux of 5 times 10 to the negative 5 milligrams per second. This explains why the original drug was failing. It simply was not crossing the barrier in large enough quantities to be medically effective. To prove that chemical modification can solve this problem, we will calculate the efficacy of a newly modified, highly lipid-soluble version of the drug. By changing its chemical structure, we increase its permeability coefficient to 5 times 10 to the negative 6 centimeters per second. The surface area and concentration difference remain exactly the same. We substitute this new, higher permeability into our flux equation. Multiplying 5 times 10 to the negative 6 by 50 and then by 10 yields a new flux of 2.5 times 10 to the negative 3 milligrams per second. This step establishes the raw delivery rate of our improved medication, setting us up to mathematically prove just how much better this new design performs compared to the original. Now, we will conclude our proof by comparing the new drug delivery to the old one. We do this by setting up a ratio of the new flux divided by the old flux. We substitute our calculated values, 2.5 times 10 to the negative 3, divided by 5 times 10 to the negative 5. When we divide the numbers and simplify the exponents, we get 0.5 times 100, which equals exactly 50. We have mathematically proven that the new flux is exactly 50 times greater than the old flux. By understanding the mathematics of the blood-brain barrier, we successfully modified the drug's chemical properties to bypass the strict security, proving how crucial permeability is for treating neurological diseases. We have covered a lot of ground today, so let us reinforce the most important ideas. First, the blood-brain barrier is an essential, highly selective security system that protects the delicate environment of your central nervous system. Second, this impenetrable seal is made possible by the tight junctions between endothelial cells, actively supported by astrocytes. Third, while it brilliantly keeps dangerous toxins out and lets vital nutrients in, it creates a massive challenge for doctors trying to deliver medications. Finally, as we proved mathematically, understanding the physics of permeability is the key to bypassing this barrier and saving lives. You now have a solid foundation in one of the most critical topics in neuroscience. Great work!